Hello, everyone. I'm Cedric from MDPI, and I will be your host for today. Welcome to the first live session of the first International Electronic Conference on Entomology, IECE 2021. This e-conference is hosted online from the 1st to the 15th July 2021 and is organized by MDPI and the journal Insects. During this time, the conference presentation will be available for discussion on the conference website. I would like to thank the conference chair, Professor Nickolas Kavallieratos, the conference committee and session chairs for making this e-conference possible, along with the speakers and, of course, our attendees. I would also like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the conference website. Today's session is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us on there. Today's live session will focus on the first three topics of the conference. There will be 5 presentations of 20 minutes each, and each presentation is followed by a short question time moderated by the session chairs. If you have any questions for our speakers, please feel free to post them in the Q&A box, which you can find in the Zoom menu bar at the bottom of your screen. This e-conference is chaired by Professor Nickolas Kavallieratos. He is an assistant professor of agricultural entomology and archaeology at the Agricultural University of Athens in Greece since 2016, while he served as a lecturer at the same university between 2014 and 2016. He has also worked at several universities and institutes in Greece, Europe, and Africa since 2000. And since 2009, he has been a frequent visiting scientist at the United States Department of Agriculture. He has published more than 250 papers in peer-reviewed journals and is associate editor-in-chief of the journal Insects, associate editor of Entomology Generalis, and subject editor of the Journal of Insect Science. He also serves as a member of the editorial board of several international peer-reviewed journals and coordinated 30 international and national projects, while he was or still is a member of the scientific team of 25 international and national projects. Professor Kavallieratos contributed to the development of several products that became commercially available through the aforementioned projects. So thank you very much, Nickolas, for chairing this e-conference. I now hand over the mic to you, and I hope you all enjoy. Thank you.
Thank you so much. Can you hear me normally?
Yeah, we can hear you.
Okay, thank you so much for the wonderful introduction. So, colleagues all over the world, it is my great pleasure to welcome you to the first International Electronic Conference on Entomology. Several months ago, Barbara Wang, representing the journal Insects, proposed me this idea, which sounded both challenging and interesting. Thus, step by step, we reached this second day of the first IECE. I was used to seeing audience during opening ceremonies of former conferences instead of speaking in front of a PC. The current circumstances still keeps us far from each other, not for long, I hope. Fortunately, technology is a valuable tool that helps us to stay in close contact and communicate our most recent findings upon entomology, which, as everybody knows, is a pure Greek word. This new approach to entomological conferences is not temporal, but it will be continued. That's why we named the first IECE, the second, and the third IECE, and so on will follow. The reasons are very simple. We are far, but close enough. We don't travel, but we see each other, and the most important, this first electronic conference is very successful. Here are some statistical data that encouraged us to establish a series of IECE. We received contributions from 708 people from all continents, representing 51 countries. We received 236 submissions. We accepted 178 submissions. We are now publishing 130 full papers and 48 posters. Our main philosophy was based on the global collection of full papers and posters that would be previously reviewed. At this point, I would like to thank all scientists who supported this very first effort. I would like to give a big thank to our session chairs, who worked very hard during the evaluation of abstracts and full papers. There is some extra work to moderate the sessions, but I'm pretty sure they will manage everything perfectly. Although you have seen their photos, I would like to name them, to hear their names. Angeliko Petrovic, University of Belgrade, Serbia. Erin, Scully, USDA, USA. Mihailo, Jelić, University of Belgrade, Serbia. Kristopher, Giles, Oklahoma State University, USA. Blas Lavantero, Universidad de Talca, Chile. Donato Romano, Santa Ana School of Advanced Studies, Italy. Antonio Onofre Soares, University of the Azores, Portugal. Rob Morrison, USDA, USA. Brinkton Mvumi, University of Zimbabwe, Zimbabwe. Jerry Zhu, USDA, USA. David Coyle, Clemson University, USA. Dimitrios Avgis, Agricultural Organization, Demeter, Greece. Andre Wilke, University of Miami, USA. Silvio Erler, Julius Kurn Institute, Germany. Ljubica Stanisavljevic, University of Belgrade, Serbia. The same stands with our conference committee. Giovanni Benelli, University of Pisa, Italy. Barbara Mantovani, University of Bologna, Italy. Andrew Polazek, Natural History Museum, United Kingdom. Maurizio Francesco Brivio, University of Insubria, Italy. Gabor Lovey, Aarhus University, Denmark. Emmanuel Roditakis, Hellenic Mediterranean University, Greece. Rosemary Collier, University of Warwick, United Kingdom. Dionysios Perdikis, Agricultural University of Athens, Greece. Barbara Conti, University of Pisa, Italy. Mariana Prazic-Golic, Institute of Pesticides and Environmental Protection, Serbia. Goran Andrić, Institute of Pesticides and Environmental Protection, Serbia. Joan Corley, IFAB, Argentina. Brett Harley, University of Pretoria, South Africa. Ferenc Lakatos, University of Sopron, Hungary. Natalia Kirichenko, Siberian Federal University, Russia. Guido Favia, University of Camerino, Italy. Yuval Kodlieb, the Hebrew University of Jerusalem, Israel. And Guy Bloch, the Hebrew University of Jerusalem, Israel. Many thanks should be also given to our invited speakers. Neil Audsley, Newcastle University, United Kingdom. Guy Bloch, the Hebrew University of Jerusalem, Israel. Paulo Borges, University of Azores, Portugal. David Coyle, Clemson University, USA. Guido Favia, University of Camerino, Italy. Peter Kevan, University of Guelph, Canada. Claudio Lazzari, University of Tours, France. Fabrice Legeai, NRAE, France. Ferenc Lakatos, University of Sopron, Hungary. Mara Lawniczak, Wellcome Sanger Institute, United Kingdom. Angeliko Petrovic, University of Belgrade, Serbia. Andre Wilke, University of Miami, USA. My special thanks should go also to the team of insects who real exhaustively worked the last several months. Miss Fancy Zai, Miss Betsy Feng, and Miss Barbara Wang. I'm also thanking insects for providing the organization of this event and the journals Agriculture and Biology for being sponsors of the first IECE. Last but not least, I'm waiting your full papers for potential publication in Insects after passing the normal reviewing process. It is my great pleasure to announce a remarkable increase of the impact factor of insects, which now is ranked 18th among 102 journals in category of entomology. But I think it is time to move to our first session. Thank you, all of you, for your attention and your precious contributions.
Thank you. Yes, as you just said, we will start with our first session, actually session 2, entitled Genetics and Genomics. And it's my pleasure to introduce briefly the session chairs of this session to you all. Dr. Erin Scully from the Center for Grain and Animal Health Research in Manhattan, Kansas, in the United States, and Dr. Mihailo Jelić from the Faculty of Biology from the University of Belgrade in Serbia. Thank you both for accepting to take part in our conference, and I now hand over the floor to you to start the session. Thank you.
Thank you. I would like to thank the organizers for letting me in. It is my great pleasure to announce our first speaker. She is Mara Lawniczak. She is an evolutionary geneticist and senior group leader at the Wellcome Institute. Current malaria research in her group spans vector population genomics and parasite transmission biology. Her team is generating high-quality reference genomes from single mosquitoes and sequencing thousands of wild mosquitoes. Her group also created and continues to build the Malaria Cell Atlas. She examines individual parasite transcriptomes in natural infections to investigate transmission dynamics. Mara, please give us your presentation.
Thank you very much. I will share my screen. So thank you very much for having me. I'm going to talk today about a targeted sequencing panel for malaria control and biomonitoring. The picture that you can see here on the screen is a photograph taken by Alex Wild of an Anopheles mosquito, and there are more than 200 million cases of malaria every year and nearly 500,000 deaths of usually small children in Africa every year. And every single person that's got malaria or that has died from malaria has in common that they've been bitten by one of these mosquitoes. The malaria parasites rely entirely on these mosquitoes to get from human to human. And it's this fact that has actually made vector control really the mainstay of malaria control. So the Anopheles genus is actually a very old genus, and when these ancestral species evolved, they probably did so at a time when South America and Africa were still touching. There are more than 500 species in the Anopheles genus, broken down into 6 or 7 groups, of which 4 have major malaria vectors, and those are listed here. These are subgenera that have, or subgroups that basically have varying amounts of species diversity. About 10%, so 50 species of the known 500 species in this Anopheles genus are able to transmit human malaria. And in fact, this is the only group of mosquitoes that do transmit human malaria. Other mosquitoes can transmit avian malaria and other types of malaria, but human malaria is only done by Anopheles mosquitoes. So as well as having very highly diverged species in the group, there's also some complications within species groups. So in this particular group, Pyrotophoris, for example, I'll talk about that next, sits the Anopheles gambi species complex. So Anopheles gambi is far and away the most famous of the Anopheles vectors that transmit malaria, and that's because it is one of the most dominant mosquito vectors in Africa, where the vast majority of malaria mortality and morbidity is happening. These species are all found in Africa, and they are all morphologically identical, but they vary quite a lot in terms of their ability to transmit. So Calitzi, Gambi, and Arabiensis are all considered major malaria vectors, and they are widespread across the continent. The rest of these are much more focally located in different geographic regions of Africa. And Melis and Maris are saltwater breeders, or brackish water breeders, but they can transmit and they contribute to transmission locally, but because their geographic range is actually quite restricted, they're not major vectors. So it's complicated because these all look the same. And so you can't look at a mosquito and know this is a mosquito, this is a malaria vector. And some of these don't even bite humans at all. It's a big question. How do we really target these mosquitoes and understand what their biology is all about if you can't see the difference? So of course, there's molecular tools to do that. Like I said, so the Gambi complex that I just gave you a feel for in terms of how the complications there around identifying vectors and non-vectors, that's also true for the Funestus group, which is another species group, another major malaria vector in Africa. So there's basically 4 major malaria vectors in Africa: Gambi, Coluzi, and Arabiensis, which are all part of one group, and that they can interbreed, and so they can share alleles through them, which is why they're called a species complex. And then Funestus, the Funestus group actually has major vectors, but also many non-vectors in that group that are morphologically very similar and difficult to distinguish. Now, if you're trying to study them, like I said, there are these molecular tools, but of course you kind of have to know what you're looking at in order to employ them, because you have to typically, when you're working in a species complex, these sort of traditional cytochrome oxidase markers don't work very well to identify to species level. So each of these different groups has had its kind of own bespoke set of primers created, where you can sort of have, you can run a simple PCR using conserved primers that amplify, perhaps, for example, across the entire Gambi complex, and it will tell you, based on the size of the band in your gel, this is Gambi, this is Arabiensis, this is Quadriannulatus. You have to know what you're looking at to get a band, right? So you have to know you've got a Gambi group individual. So when you get a failure to amplify, you never really know, is that because I identified it to the wrong species complex and employed the wrong primers? Is it because my DNA is bad? Or is it because something that is in this complex but doesn't actually behave well according to the primers I have? So these traditional markers like cytochrome oxidase don't work well, and the diagnostic markers that do work well still don't give you clear resolution on things that don't give you an expected band size or an expected band at all. So we think that this is a very difficult, this is a really important problem to crack, to try to create a tool that actually will give you all that information in one go. And sort of what species are you working on at any level of the full phylogeny of Anopheles, but also give you detailed information on when you're working in a species complex, which exact species are you working on? This is important because we are targeting mosquitoes massively in order to halt malaria. And the species ranges are changing rapidly because of the climate crisis and because of vector control initiatives too. So on top of that, our other control initiatives also influence the biology of the organisms that we're going after. So for example, all the bed nets and all the indoor residual spraying that happens to target mosquitoes, these major malaria vectors that I've mentioned that tend to go indoors and bite people while they're sleeping at night. Well, that puts a lot of selective pressure on them to start biting outdoors or to start biting earlier in the days. So that causes behavioral changes. We need to be able to have ways of monitoring that very carefully. When you have a behavioral change and you start to still see transmission, even though there don't appear to be indoor biting mosquitoes anymore, is that because it's a different species transmitting, or is that because the species you were targeting has evolved away from your control efforts? So this is the foundation of why we set out to create this kind of common language tool that operates across the entire Anopheles genus, but also goes deep into the species complexes and even tells you sort of within a species, which geographic location did it come from. So we've created a multi-locus DNA barcoding panel. And so this is targeted, short 200 base pieces across the entire nuclear genome of the kind of generic Anopheles mosquito. And the way we were able to do this is because Anopheles is very a rich genomic kind of species, because we have more than 20 reference genomes that have been around for some years. So you can do those kind of multi-genome alignments and identify across the genome sites that are very highly conserved, close to each other, that would be good primer binding sites across the entire genus, but still contain in the DNA in between them something that gives you some sense of what you're looking at. On top of wanting to know what the species is, we also want to know, are they contributing to malaria transmission? And so we also added into this panel 2 generic Plasmodium mitochondrial loci that tell you, is this insect carrying parasites? Doesn't tell you anything about whether it's transmitting them, because you have to know exactly what you could actually be targeting a mosquito that bit a human but was unable to transmit those parasites onwards. So you need to know something about this sort of the tissue you're looking at to be confident that you've got actual transmission happening. So this is the kind of schematic that we've got. And these were two postdocs in my group who really worked this out. Anopheles albuminus is one of the most highly diverged species from Anopheles gambiae. We think there's about 100 million years of evolution in between these. And so what you can see here is a schematic of the chromosomes, the chromosomal arms of the Anopheles, the general Anopheles genome. And if it's red, then it's a diagnostic, it's a targeted amplicon that sits inside of an exon. If it's yellow, it sits inside of an intron, and if it's blue, it's intergenic. And so you can see that there's lots of rearrangements within chromosomes, but actually across the chromosomes, there's a lot of conservation in terms of where these amplicons are located. And this is just showing you 3 of the species, but we actually did this. We did this multi-genome alignment for all of the species that have reference genomes available. And so we have this kind of schematic for other organisms too, but most of the other species don't have chromosomal, clear chromosomal assemblies yet. So it's harder to pin them to the chromosomes. Another big part of our efforts was to try to figure out a way in which we can do this at massive scale, because it takes a lot of time for each individual mosquito to look at it morphologically, to extract its DNA, to run a PCR. And so we wanted to do this in plates with actually all you need to do is look at it to know it's an Anopheles mosquito. You don't need to take it to its species or its group even. And we have a very cheap extraction approach now. That's our paper actually just came out, I think last week. That's present in this paper. And what this allows us to do is preserve the morphology of the specimen too, because we expect that we will see things that we did not expect. New species or weird species in places that we did not expect them to be. And we want to be able to return to the specimen and say, does this really fit with the morphology that we do understand? So this is a schematic of just what we do. We actually put all the individual insects, 1 per well of a -6 well plate. It's a very simple, cheap lysis buffer. And then we long-term store the samples in the plate still. We take a stamp of the lysis buffer and we do a 1 in 10 dilution, and that is the input material for the multiplex PCR. So each of those individual DNAs then goes for a single PCR that has 64 primer pairs in it, 1 PCR. Each of the primer pairs has a UMI, a unique molecular index kind of code attached to it. And so you know the origin of the mosquito when you sequence the DNA that it came from well A1, all 62 loci of the mosquito and 2 loci targeting the parasite. So what this allows us to do is sequence about 1,000 individual mosquitoes on a single MySeq run. And this is just a schematic to show you kind of this, what we're doing, what we're looking at. These are not mosquitoes, these are flies caught in a different project, but basically they're just sitting in a well, releasing their DNA non-destructively into the lysis buffer. So we've now pushed more than 40 species through the actual panel. So we've got DNAs from across the world, from partners in Southeast Asia and in South America and in Africa. And what this tells you on the y-axis here is the number of amplicons of those 62 that we recovered from these different species that are located here on the x-axis. And these are showing you the different groups that I gave you a sense of in the beginning. And you can see the more highly diverged groups from the Anopheles gambiae group are returning fewer amplicons worth of data, but still we tend to get more than 30, and that's plenty to assign a species. This just happens to be because we had higher quality genomes from this end, so we were biased towards recovering amplicons from those species. The other thing that we know now from the data is that the amplicon data, even though on average it's only 10,000 bases per individual mosquito compared to the 250 million bases that make up the genome. On the right here is the whole genome alignment from the species that have reference genomes. And on the left is our recreation of the phylogeny from the amplicon data only. And you can see these really pretty well mirror each other. And we even can get some resolution into things that we know are species groups that are very challenging to work on, like the Custani and the Gambiae and the Marshalli group. But these are not particularly well supported. So there's more work to be done here in terms of gaining confidence, which I'll talk about next. We have a project that I won't talk about at all today, which has been going on for quite a few years now, called the Anopheles gambiae 1000 Genomes Project, This is an open data resource, so you can go and look at the data. There's a really wonderful genome browser called Panoptes, and you can see 765 individual Anopheles gambiae and Coluzi mosquitoes that have been sequenced, wild caught across Africa, sequenced, and their whole genomes are available. This gives a reflection of the population structure that we in silico created. So we pretended for those 1,000 genomes that we've already sequenced and released the data, that we had, instead of doing that, amplified it using our amplicon panel. So this is just using the genome sequence that would have been amplified through those 62 primer pairs. And what you can see is that it faithfully reproduces population structure and species. So the circles over here on the left are Coluzi, and the X's over here on the right are gambiae. And these are species that are extremely closely together genetically. So they are very hard to actually differentiate. They tend to be differentiated by a diagnostic primer in the X chromosome, where the ribosomal DNA locus is. And that's, otherwise it's very hard to tell them apart. So the panel is doing a very good job at differentiating them. But we want to have a clear kind of classifying approach for how we, when new samples come in, how do we assign them to a species? And this is work we're doing now with a PhD student in the group, Mihailo Baudet. So she's developed a two-stage hierarchical classifier. And what's exciting about this is that it uses k-mers. So instead of trying to do alignments of those 200 bases across a very divergent set of species where the alignments would look very trash, like really garbage, we use k-mers, which allows us to avoid alignments, and all those indels that happen inside of those don't throw us off, and they can be highly divergent. So for every amplicon in every sample, she basically takes a k-mer pool for that amplicon and asks what's the nearest match to our labeled reference data set using the samples that we got from partners who carefully morphologically identified and told us what the species was. We extracted the DNA, we put it through the panel. So we have this kind of index of this is known, these are known amplicons from these set of species. And now we're just trying to match our new samples going through to that. So it was basically perfectly accurate, this approach. The two errors that you see here where there was a mismatch, you can see these in the bars of color. Actually, it turned out when we went back to look at the cytochromoxidase information that it was a misidentification of the original specimen to begin with. So how does this work in practice? This is an example. We had a set of 600 mosquitoes sent to us from a collaborator in Nigeria. Nigeria is a very important gap in our genomic data set. It's a major malaria transmission area, and we don't really have a great view on the genomic diversity of the mosquitoes there. These were all caught indoors as part of a trial studying ivermectin, and they were quickly morphologically identified to all be Anopheles gambiae. When we put them through our panel, actually, we discovered only a third of them looked to be Anopheles gambiae, and the rest looked to be Anopheles funestus. And we confirmed that with these alignments here, just showing you a sense of the divergence between down on the bottom here, the funestus individuals, and up at the top, the Arabiensis, Coluzi, and Gambiae individuals, which are all very closely related. So effectively, it's working to get you to the species level, and then the second to the species group level, and then within species group where you have some introgression happening between species, then we have a second stage hierarchical classifier. And in this, we're using what's called a variational autoencoder, which is machine learning, deep learning approach to binning. So it's kind of like a PCA. You're just trying to reduce this highly dimensional data down into clusters. And so what you can see here in the background that are kind of fuzzy colors is our known samples going through. And in the orange sort of dark ringed circles, you can see these new samples that are coming through that came from Nigeria, and we were able to differentiate, a handful of them being Anopheles coluzi and most of them being Anopheles gambiae and none being Anopheles arabiensis. And again, this is using a k-mer approach rather than alignments. So there are two additional amplicons in this panel that target the parasite. Mosquitoes, they blood feed, and the parasites take about 24 hours to do a little bit of reproduction in that blood meal and then invade the gut of the mosquito and start to form this kind of wart-like sac on the outside of the midgut, where they develop over the course of about 10 days before they then invade the salivary glands, and then the mosquito is infectious again. So it's kind of remarkable, but it takes over 10 days for a mosquito to go from feeding on a person who's infected to actually being infectious itself. So that's what's represented here on the bottom is studying these mosquitoes. So we fed them in the lab. And of course, if they feed on blood that's infected, there's loads of parasites in there, so we detect them. And then some days later, some of the mosquitoes get infected, and those are up here, and some of them don't. And here's our uninfected controls. So we know that this mitochondrial target is working to tell us whether the mosquito has evidence of the parasite in it or not. So what's next? Over the next 5 years, we are carrying out a massive endeavor to try to apply this tool to 500,000 mosquitoes from across Africa. We think we're really interested in individual level mosquitoes. So to tell us something about, what species is it? What's its population structure in comparison to many other species, many other samples from within its species, but also is it transmitting, is it carrying parasites? However, although that's our primary interest, we also think that this panel is great for studying larval breeding sites, for example, which is a big question. You could use eDNA to put through the panel to understand what evidence there is of these different mosquitoes present in the breeding in that water, or bulk samples from traps, for example. The non-destructive extractions that I presented, I think should be really relevant to people attending this conference who are interested in studying their organisms from a genetic and morphological perspective, and also it's really cheap. I didn't give you the results, but actually those DNAs are viable for whole genome sequencing and give you plenty of DNA. It's a high throughput and low cost protocol. And the thing that we think is most important is that it's fixing all these little problems I told you about in the beginning, where people are working in bespoke primer sets and unable to kind of understand the differences. When they see failures, they don't know what the origin of the failure is. So it's this common language diagnostic. And it will help to identify new species, we think, too, and contribute our understanding of what species are actually transmitting. Malaria mosquitoes are a major target of human interventions, and we're trying to push them down this path to eradication, but what we always do is push them down this path to resistance. And so by doing kind of long-term biomonitoring, hopefully we'll be able to understand how to do a better job of getting them down this path towards eradication. This is my second to last slide, and I just wanted to give a sense of other things. So this is the project I've just told you about, the 100,000 anophelines a year project that we're carrying out. At a sort of bigger investment per individual scale, computational wise, but many fewer individuals, we're doing two other projects. One is around continuing our Anopheles gambiae 1,000 genomes style project, where we're studying mosquito population genomics at a whole genome level across Africa, where there's ongoing intervention efforts going on in space and time. And then also, we have been really working hard to do single mosquito reference genomes for all of the Anopheles species. We're just at the very beginning of this project, but it's going really well. We've generated 20 different vector species reference genomes, which should be released within the next few months. And these are higher quality reference genomes than we've ever had before, because of some of the technological improvements and the ability to go down to a much lower input material amount. So we can do this now with a single mosquito. And with that, I will end and just say this large-scale study is going to depend heavily on partners who have long-term sites where they're studying malaria vectors. So if that sounds like you, please get in touch with us. We'd be really interested to talk about your projects and whether this panel would be useful. We have funding to push a lot of mosquitoes through it. Over time, we hope to make the actual panel available to be run in country. It's going to take us a little bit of time to do that. And I will end there and just thank my funders.
Thank you, Mara, for this inspiring lecture. Now we have several minutes for questions and answers.
I can get us started off with a quick question for Mara. Based on the classification panel that you sort of developed, how many species are showing clear signs of being able to be classified to the species level versus those that are sort of showing ambiguities? You mentioned the potential to identify new species with that amplicon panel. So I was just curious, I know you just got started with that. What are you seeing, and how many appear to be new species?
I think in order to be confident that we're seeing something that is distinct and representative, we need more samples because we have pushed very few individuals for each species through so far. We're talking like 3 to 10 individuals typically, with the exception of Anopheles gambiae and Anopheles funestis groups, where we've pushed more than 1,000 individuals through. So I can really only give you a sense with the gambiae group that we know what gambiae is and we know what Galuzzi is. They've got diagnostic primers that we can basically use to kind of understand the clusters that are Gambi and Kalutsi. And I didn't get into it here, but we actually see a cluster in between the two of them, which is all West African. And there's some confusion about what these West African individual mosquitoes are, because they look like Gambi in the diagnostic, but they actually look like Kalutsi in their genomes. So it's still being worked out exactly, is this a new species or a different species, or is this some kind of weird hybrid form, or is this you know, a hybrid collapse, or what's going on there? So I think what's clear is that we see this cluster in the output in that picture. You know, there's like a clear cluster that sits in between, and so it identifies a set of individuals that we need to go after in a genomic fashion to understand, are they different genomically? And so I don't have an exact answer for your question, because we don't have enough individuals that have gone through, but I suspect that same sort of thing will happen. Once we start to get hundreds of individuals of the different species, we'll start to see clusters that don't have a name, that are clearly different, and then we can target those.
Thank you, that's very interesting.
Okay, thank you. I don't see any more questions, and I would like to ask Mara whether they ever analyzed the apicoplastic genomes of parasites, and can they tell you anything?
That's a good question. I don't know the answer. I don't even know how big the genome is of the apicoplast. So it's not one that we've targeted in our panel. I think that there's data for it, but not my area. Sorry.
Okay, thank you. Okay, we can proceed further.
There is one question in the chat from Amir Zaman Shah. I don't know if it's complete. I just see part of it. What's the most important and initial measure to avoid something? You see that?
Yes, yes. To avoid.
Oh, here it goes. You typed the full one. There we go.
To avoid initial infestation of dengue mosquitoes. What is the most important initial measure to avoid initial infestation of dengue mosquitoes?
Outside of my area of expertise, I don't work on dengue mosquitoes, but I would say larval breeding sites are often a big issue with Aedes mosquitoes, because they can breed in very small puddles of water that probably contribute a lot to dengue transmission.
Okay, are there any more questions?
I don't see any more.
No, I don't see too. Then Erin, please proceed. Thank you, Mara.
Okay.
Bye.
Thank you. All right. It's my pleasure to introduce the next speaker for this session, Dr. Fabrice Legeai from the INRAE. There we go. Fabrice is a project manager, an expert in bioinformatics, and also a group leader. Since 2007, he's been managing the bioinformatics platform for the arthropods of ecosystems, gathering genomics data from more than 35 different insect species. He's worked on aphids, Lepidoptera, and also parasitic wasp genomes. And from that, he's been able to develop various workflows and analysis for genome assembly, annotation, differential expression analysis, predicting and the identification and analysis of variation among populations. He's participating to develop softwares for omics data integration, including AskOmics, differential expression analysis, which is AskOR, which he will be speaking about today, long non-coding RNA prediction, and genome gap filling and structural variant prediction with linked reads. So with that, I'd like to turn it over to our speaker. The floor is yours.
Okay, many thanks for the presentation. I will share my screen. You let me know if you can see it correctly. Okay, thanks a lot for the invitation. And let me introduce my work about AR package for the analysis of RNA-seq data, AR package as we call AskOR. Okay, so you already introduced myself, so maybe I won't be that long, but by the way, I'm working on designing standardized protocols for supporting economical insects in France at NRAE, which was called INRAB previously, but it's dependent from the agriculture department in France. So at BIPA, we are developing such protocols, but also we are offering a web portal for collaborative analysis of genomes at the address, which is written here, bipa.genwest.org. So at this address, we have already 39 available genomes, mainly aphids, but also parasitoids, wasp and Lepidoptera, and also insect from other orders. And on this web portal, you can find many tools, various tools. Some are gene reports, blood search, genome research, genome browsers, search engines where you can find your favorite genes, some tools for looking for the expression of the genes in many different tissues, and tools also for comparing the synteny of genome, different regions of genome from different genomes, from different organisms, and also identifying duplication or looking for duplication in many different gene families in phylograms. And finally, we also offer, we are hosting genomes, and we are doing genome annotations and assembly annotation, and we also allow the curation, the manual creation of genome until the release of official gene sets. And during all that process, everything could be public, but we can also work within a restrictive access with consortium upon request. So at our website, we also deliver a Galaxy server. Galaxy server is famous tools that allow people to use many different software for working on bioinformatics, for working on genomics, and doing data analysis directly from your web browser instead of using command lines. And through BIPA, you can directly have access to computing cluster, and where 3 nodes are dedicated to Galaxy with almost 200 CPUs and more than 1 terabyte of memory. So in such Galaxy, you can run standard, usual procedure for analyzing RNA-seq data. And the first part of this analysis concerns the mapping on different references. If you have already a genome, you can directly map on genome and the annotation on genes. If you do not have a genome yet, you can directly map your reads against a reference, which can be done by others. And if you do not have any reference, even a transcriptome reference, you can also do an assembly of your reads and perform your mapping on that assembly. So that part could be easily done through Galaxy, through the Galaxy server that we are proposing at BIPA and many other Galaxy server on different bioinformatics platforms. But the second part of the RNA-seq data analysis concerns the statistical analysis to identify genes that are differentially expressed between different conditions. And sometimes it's hard for people that are not very fluent in R or that are not used to programming, just to work with the data and just to use different R libraries. It's why with people, with collaborators, we decided to develop a pipeline just for dealing with such RNA-seq data and doing the statistical analysis as straightforward as it is possible. So this is the aim of ASCOR, and ASCOR is just starting from raw counts that has been calculated by the process just I told before. And with that ASCOR, you can check the consistency of the sample, you can normalize your data, you can identify the differentially expressed genes between contrast, and you can also perform some gene ontology enrichment and do expression profiling. So you can do that directly by downloading ASCOR into your favorite R environment, which could be a studio, but you could also work through a Galaxy portal like the other tools I told previously. For instance, when you run Galaxy, if you are familiar to that interface, on the left panel, you have a lot of different tools, and there you can find ASCOR, RStudio environment. You just have to run it, and you go directly on your web browser. You have directly access to an RStudio server there, and you can directly start to run with Ask.OR. It's very easy. You can directly load your data from Galaxy uses single commands, and you can also download the data from the RStudio server to the Galaxy server with also a single command. So to go back to Ask.OR, what it is, how it is working, so it is very simple. Well, we try to do it as simple as it's possible. And first, you just have to put some data in a directory, and then you just have to run a script. When we are delivering, we are providing a template for that script, and you just have to modify some parameters. Then you run sequentially 12 functions, and at the end you get some multiple directories with a lot of output, which can be data that could retrieve into that you could download to your computer, or graphics, or many graphics. So for the input file, you just have to give the raw counts, sample descriptions, just indicating your samples, which condition it came from, and see optionally gene annotations or assignation of the gene to the gene ontology. You have also to provide a contrast. So the contrast you have to perform, and for providing the contrast is quite easy to indicate in a single file, in a single tabular file. I put an example here. For instance, if you have 3 treatments and 3 time points, you just have to consider all the conditions, the 9 conditions of your contrast, 9 conditions that you have sequenced, even if you have 4 replicates. Then if you need to compare the first time point to the 2 other time points, you just have to put a plus sign in the first time point and minus sign to the other time points. The time points you would like to compare to. For instance, if you like to compare the time point 1 to the time point 2, in the table you just have to put a plus for the A1, B1, and C1 conditions, and a minus to the A2, B2, and C2 condition. And it's exactly the same for the 1 versus 3. And you can do that even if you are comparing one time point to the two other time points, just to put a sign in the same way. And even if you can do any contrast you like to do, for instance, if you like to compare the treatment C to treatment A, you just have to fill the form, fill the table like that. Then you can start by doing the statistical analysis, first by removing the low expressed genes, applying very simple rules, which keep the genes with the CPM values higher than the threshold for minimal number of samples. And you can normalize your data using the standard TRIMM mean values, TMM, which is provided by Edger, but you can change it to the relative log or upper quartile normalization. Then you can get a correlation, a parallelogram that indicates the correlation between your samples. Using this parallelogram, you can directly figure out if your samples, if your replicates are well, this correlation between your replicates is okay. And you have also a multidimensional scaling plot where you can identify outliers or maybe samples with erroneous samples, so that you can remove for further analyzing and continue without into Ask.OR, continue without such sample because they are bad. Then you can perform the usual differential expression analysis still using Edger, doing the estimation of the dispersion first, then generating a general linear model, GLM, with different methods. Then you get, for all the constructs you have performed, you get the number of genes that are efficiently expressed and a table indicating with the value, all the genes and the p-values, the FDR, log-fold change corresponding to the test. And at the end, also a summarizing table that give you a number, which is 0, 1, or -1, indicating if the genes is over or under expressed in some of the tested conditions, some contrast. We end with the annotations that you have provided at the beginning of the process. Then using such files, you can directly use ASCOMIX, which is a tool that we are also developing that helps to integrate those data all together and to compare it with other data coming from different data sets. For instance, you can select genes that are upper expressed in some conditions and lowly expressed on other conditions, and see if the genes are targeted from some microRNAs or if the landscape, epigenomics landscape of these genes are very particular. So, but if you are not used to ASCOMIX, if you do not want to use ASCOMIX, we are also giving you access to some usual Venn diagrams that is set from a select list of content helps you to see the intersection of all your contrasts you have already done with ASCOR. And if you have more than 3 or 4 contrasts, we also give some upset graph that is very useful and very readable for seeing the intersection of the contrasts. There you can see is the gene is the number of genes that are overexpressed in some contrasts and are also underexpressed in other contrasts. We are also providing tools and functions that helps you to see if the contrast is the genes that are derived from that contrast are enriched in some genetology term. So you have a list of terms for each contrast, a list of terms, and the p-value indicating if that term are those terms are overexpressed. And you also and finally we also give tool to do the expression profiling of the genes. It means that you can see if some genes are share the same profile of expression between conditions, even if you are not working on the kinetics. So we are using COSEC for doing that, and COSEC has the advantage to choose the base case, the base number of cluster using your data set, and also give you some indication about the robustness of that clusters. It means that the number of genes that could be associated to another cluster if you redo the analysis that appears in red in that bar plot. So we are also providing at the end a lot of different figures that you could directly use into your publication. And we also give a genetology arrangement for the cluster, and we also provide upset graph that helps you to see if the expression profiles, if the cluster obtained by expression profiling are similar or not, or including some genes that are already found in the contrast analysis. So finally, we had a recent, we had also a recent new graph and recent functionalities which give you for a list of candidate genes a lot of information directly into a graph that where you can see if the genes you selected are belonging to a cluster or if they are not expressed, and a heat map showing the expression in the different conditions you are testing, as well as a point give you an overview of where are the what are the conditions, what are the contrasts where the genes are over or underexpressed. So as a take-home message, just would like to tell you again that BPI can support various projects about agronomical insects. So you can contact us if you have some projects you're willing to share with us. People can host genomes and offer numerous tools for collaborative analysis. And with the help of an online documentation, ASCOR is straightforward to explore RNA-seq data and produce publishable figures. ASCOR has also a pipeline which can be run easily as a single script and is very highly customizable and reproducible, and is also compliant to ASCOMIC for performing other integrative analysis. So you can directly download ASCOR at the website at GitHub here, and it will be submit to the CRAE and Biocoordinator very soon. So I would like to thank also the people working with me on ASCOMIX and BIPA. Thank for your attention.
Thank you, Fabrice. Very interesting. If anybody has any questions, feel free to either type them into the Q&A or into the chat, and we'll go ahead and give a few minutes before we need to move on to the next speaker. And I'll just go ahead and throw out a question to get us started. How much data can you upload to the Galaxy server? Is there a limit per individual as to how much RNA-seq data somebody can put on your server? Just curious.
By the way, we have some quota. I don't remember exactly how much it is, but you can add to that. We put you a higher quota if you
okay.
are willing to. But by the way, we cannot give access to anyone with a lot of hard disk as well. So yeah, I cannot answer you exactly the quota now, but I could check.
Mm-hmm. Thank you. Any other questions for Fabrice before we move on? Saloom Azizi says, thanks, Fabrice. This will make life easier. I totally agree with that. It's, learning the programming is probably 80% of, what you need to do when you analyze RNA-seq data. And so tools like this are very important for help making these types of analysis accessible for all. Okay, well, seeing no other questions or comments, thanks again, and we'll go on and move to the next session. Thank you all for attending, and thank you to the speakers for their wonderful presentations.
Thank you very much, Erin. So according to the program, we will now have a short break. So see you in 10 minutes. All right, welcome back. The next session is called Biology, Behavior and Physiology, and is chaired by Professor Kristopher Giles from the Oklahoma State University in the United States, Dr. Blas Lavandero from the Universidad de Talca in Chile, and Dr. Donato Romano from the Biorobotics Institute and Department of Excellence in Robotics and AI at the Sant'Anna School of Advanced Studies in Pisa, Italy. Many thanks to all of you for sharing this session. The floor is yours. Thank you.
Yes, I will present Dr. Neil Audsley. Dr. Neil Audsley from School of Natural and Environmental Sciences in Newcastle University. Dr. Audsley is an invertebrate physiologist and senior lecturer at Fair Science and Newcastle University. Neil Audsley provides strategic research and development and policy advice for the management of native, invasive, and emerging pests of agriculture, horticulture, and tree health importance. His major research interests include invertebrate endocrinology, physiology, and biochemistry, novel pesticide targets, insecticide resistance, integrated pest management, and biological control. He is currently leading projects on the biological control of invasive pests of tree health importance and developing alternative strategies for the control of insect pests by targeting insect neuropeptides and their receptor. So looking forward to hearing this interesting presentation from Dr. Audsley, and the floor is yours.
Thank you. I'll just share my screen. Okay, is that now? I presume that's now shared.
Yeah, perfect. Thank you.
Okay. Okay, well, thank you very much for that introduction. I currently have a joint appointment actually with Newcastle University and Ferrer Science Limited, and I'm currently based at the Ferrer Laboratories in just outside York in the UK. So today I just want to present my work I've been doing over the last few years on the control of feeding behaviour with insects and their practical implications. So as in most animals, feeding is very complex, involving positive and negative sensory mechanisms from peripheral systems, such as stretch receptors, from hemolymph composition, and from hormones. On feeding, a cocktail of peptides are released both centrally from the brain and locally via the stomatogastric nervous system and the gut. And these peptides aid in the ingestion, digestion, absorption, and movement of food in the alimentary canal and in osmoregulation. In insects, the alimentary canal is made up basically of three specialized regions. The foregut, where food is stored and the process of digestion begins. The midgut, which is primarily concerned with absorption, and the hindgut and the associated Malpighian tubules that comprise the excretory system, which ultimately determines hemolymph volume and composition. For digestion and absorption to occur effectively, food is moved along the alimentary canal by peristalsis. Now, these contractions are myogenic, i.e. they occur within the muscles themselves rather than the result of nervous stimulation, and occur predominantly in the foregut. And the movement of food is regulated by the proventricular and pyloric valves in the foregut and the hindgut. Food uptake and transport in the foregut and midgut are regulated by the stomatogastric nervous system that consists of a number of intersegmental ganglia which have links to the central nervous system, as shown in the cartoon on the left-hand side of the slide. The precise structure of this system varies in different insect studies, but in general consists of the following. Central to this system is the frontal ganglion, which is located anterior to the brain on the dorsally to the esophagus. It innervates the buccal region of the stomadeon via the frontal nerve and is connected to the brain via the frontal connectives. A recurrent nerve links this to the hypocerebral ganglion, which has links to the corpora caudaica, which itself is an organ which synthesizes, stores and releases neuropeptides. And then the esophageal nerve links to the proventricular ganglion, from which the midgut and crop are innervated by axons and fibers from this particular ganglion. A variety of peptides have been shown to be associated with the stomatogastric nervous system. And there's a list here on the right-hand side of my slide. And many of these have a role in regulating gut motility and feeding behavior. So why am I interested in the control of feeding behavior? Well, neuropeptides and their cognate receptors, many of which are G-protein coupled receptors, have a pivotal role in the regulation and modulating physiology and behavior. They are considered prime targets for the development of insecticidal drugs, but are underexploited for this purpose. The peptide and its receptor can be identified and then manipulated to impact on physiology and behavior. Agonists can be used to overstimulate, or antagonists used to block the signaling pathway. Either way, an inappropriate physiological response ensues, resulting in death of a pest or disruption of its normal fitness or reproductive capacity. And in this way, to reduce pest populations. Functional cell-based assays can be developed to screen for lead compounds. And as mentioned in my previous slides, various peptides are associated with the stomatogastric nervous system, but I'll concentrate on two groups, the latter regulatory peptides and the myosuppressins. The alatoregulatory peptides are so called due to their ability to regulate juvenile hormone synthesis by the corpora alata. The alatostatins are inhibitory, and the alatotropins stimulate juvenile hormone synthesis. However, these peptides have multiple other functions, including the regulation of gut motility. 3 structural alatostatins, different structural alatostatins, have been described in various insects, referred to as the A-type, the B-type, and the C-type. The B-type alatostatins are also known as the myoinhibitory peptides. In insects, there are multiple alatostatin A's and myoinhibitory peptides present in individual species, whereas normally only 1 C-type and 1 alatotropin is present. We've investigated the presence of these peptides in the stomatogastric nervous system of the larvae of the tomatomorph Lacanobi oleracea. Using immunohistochemical techniques, we found that alatostatin A, alatostatin C, and alatotropin are all localized within the primary cells of the frontal ganglion. Alatostatin A is localized in both the anterior and posterior cells, whereas alatotropin and alatostatin C are only present in the anterior cells. Hence, in these particular cells, we have triple co-localization of these 3 peptides. The identity of these peptides in the frontal ganglion was confirmed using MALDI-TOF mass spectrometry of single tissues of the frontal ganglion. Of the 8 previously identified A-type alatostatins from this insect, 6 were detected by mass spectrometry of the frontal ganglion, as shown here in the masses labeled in red, as well as the alatotropin here in green and the C-type alatostatin. So looking at the biological activity, using semi-isolated preparations of the larval foregut, both types of alatostatin were shown to inhibit peristalsis in the foregut in a dose-dependent manner. Whereas alatotropin, although not shown here, was shown to be stimulatory, demonstrating their role in the regulation of food through the alimentary canal. We then went to look onto the in vivo effects of these peptides by injecting or feeding into different insects. Injections of a single dose of alatotropin or the A-type alatostatin into 5th instar larvae of Lacanobi oleracea had no effect. In contrast, a similar injection of the C-type alatostatin significantly impacted on growth and development, as shown in the slide in the center of the figure in the center of the slide. So growth was significantly impacted, as was survival, which was dramatically reduced. Most insects died within 6 days of injection, and those that survived, if they managed to proceed from the 5th to 6th stadium molt, virtually stopped feeding, resulting in death over a prolonged period of time. It's believed that the alatostatins act by inhibiting gut contractions, preventing the movement of food through the alimentary canal, suppressing feeding, ultimately causing starvation, most likely through a feedback mechanism by stretch receptors to the brain and frontal ganglion. Continually feeding alatostatin to the pea aphid nymphs over a 2-week period, shown in the figure on the right-hand side of the slide, also significantly reduced survival compared to the controls, resulting in almost 100% mortality after 13 days. We also produced a C-type alatostatin analog, which had modifications made to increase its stability. And this analog was seen to enhance both the activity and the potency of this peptide, causing death with 100% mortality within 3 days of feeding, in a similar manner to completely starving the aphids. So these data clearly demonstrate the potential for the development of a C-type alatostatin as insecticidal peptides. The C-type elatostatin receptors have been identified in various insect species, and these have been shown to have homology with the mammalian somatostatin receptor family. In insects, the receptors are expressed in the brain, the central nervous system, and the gut, supporting the role of elatostatins on gut contractions. The receptor has been characterized from the flower beetle Tribolium castaneum. A functional assay was developed by expressing the receptor in a mammalian cell line, together with a G protein and a reporter gene. As expected, the receptor is activated by the native peptide, as well as peptide analogues and similar peptides from other insects, including the elatostatin C from Manduca sexta. Its specificity was determined by testing other elatoregulatory peptides, including elatostatin A, elatotropin, and myoinhibitory peptides, all of which were inactive. Similarly, the mammalian homologue to elatostatin, somatostatin, was also inactive. The adult fly gut differs from the basic structure of the insect gut shown earlier, in that it contains this bilobe crop that is connected to the foregut via a duct and innervated by the crop nerve. Peristaltic contractions of this crop and crop duct allow movement of material from the crop to the midgut through various pumps and sphincters. Myosuppressins are conserved peptides in insects with a consensus sequence highlighted in red here. They are also multifunctional peptides and are involved in feeding and gut motility. Using antisera, which recognizes the C-terminal RF amide of this peptide, which however is not specific to myosuppressins, we found immunostaining is present in the crop nerve bundle, and nerve fibers extending over the crop surface of the invasive pest Drosophilus suzuki'i. Mass analysis of the crop nerve bundle confirms that this immunostaining is most likely due to the presence of a myosuppressin, because a mass ion corresponding to the calculated mass of this peptide was the only one detected. Using semi-isolated preparations of the crop, we also found that this myosuppressin reduces spontaneous contractions of the crop in Drosophilus suzuki'i in a dose-dependent manner, as shown in the graph. Interfering with myosuppressin signaling that regulates crop contractions is expected to disrupt the movement and digestion of food, which could potentially have insecticidal effects. However, feeding or injecting myosuppressin into fly adults or larvae had no effect. This is most likely because the arginine residue at the C-terminal consensus sequence is very susceptible to trypsin-like enzymes present in the insect gut and hemolymph, resulting in its rapid degradation and abolishing biological activity. Although only one myosuppressin peptide is encoded on the precursor gene in Drosophilus species, 2 receptors are encoded on 2 different genes in these insects. Insect myosuppressin receptors are related to the mammalian neurotensin thyrotropin releasing peptide family. In Drosophila melanogaster, these receptors are both expressed in the brain, the thoracic ganglion, and the crop, supporting its role in the regulation of crop function. Receptor assays developed to both receptors from Drosophilus suzuki'i are being used to screen peptides, analogues, and synthetic compounds to get some insights into the structural requirements for receptor activation and receptor specificity. The myosuppressin peptides and analogues tested on both receptors from Drosophilus suzuki'i are shown in the table on the left-hand side of this slide, together with the calculated EC50 values, and examples of the dose response data is shown in the figure on the right. The insect myosuppressins are pretty well conserved, as demonstrated by the amino acid sequences highlighted in red, with minor differences only seen at the N-terminus. Interestingly, the medfly myosuppressin is more potent than the native peptide on one of these receptors. And this is only due to a conserved substitution of serine for threonine at the N-terminus. The other insect myosuppressins are equally active, but much less potent, as demonstrated by their EC50 values. Deletion analogues created by removing amino acids from the N-terminus show the potency of the peptides are rapidly reduced, and removal of the first 4 amino acids from the N-terminus completely abolishes all activity. As already mentioned, myosuppressions are rapidly degraded by gut and hemolymph enzymes, with the arginine residue in the C-terminal consensus sequence being particularly susceptible. Replacement of the arginine residue with its D isomer or with an amino acid mimic to reduce susceptibility to enzymes, however, made the peptide completely inactive. But this did demonstrate the importance of this arginine residue for biological activity. The peptide was also extended at the N-terminus with a number of proline residues added to enhance its stability. This peptide was as active as the native peptide, but not as potent. Other insect peptides with similar C-terminal sequences, i.e. they contain the RF amide, were shown to have no activity on either receptors, and the related mammalian thyrotropin releasing hormone was also completely inactive. To evaluate further specificity and potential cross-reactivity, related mammalian peptides and commercially available receptor agonists were tested. Most compounds tested were completely inactive, while others were very weakly active compared to the native peptide, demonstrating little cross-reactivity between insect receptors and mammalian peptides. So in summary, feeding in insects is regulated by a cocktail of peptides released centrally from the central nervous system and locally from the stomatogastric nervous system and the gut. The foregut and midgut are controlled by the peptides within the stomatogastric nervous system. Myoactive peptides such as alatostatins, alatotropins, and myosuppressins regulate gut contractions and hence movement of food through the alimentary canal. These peptides and their receptors have been characterized and pose potential targets for the development of lead insecticidal compounds. And functional receptor assays for structure activity study and initial screening of compounds have been developed. And finally, I would like to acknowledge my colleagues at Ferroscience Limited, June Matthews and Rachel Down, and at the University of Leeds, Elwyn Isaac, Petra Bell and Caroline Goff, and to the funders for this work, who were the European Union and the Department for Environment, Food and Rural Affairs in the UK. Thank you.
Thank you, Neil. Thank you, Neil. Very clear and interesting presentation. Are there any questions for Neil? Otherwise, I have a couple of questions for you. How these cocktails of peptides are involved in affecting central pattern generators, especially concerning the gut, the peristalsis of the gut, how they affect these distributed pattern generators, if they are involved in this, of course.
Well, clearly there's a vast number of quite a large number of peptides involved, but how they're controlled centrally is at the moment is very unclear. I mean, we still know very little about the actual control and the integrated control of the gut through this this cocktail of peptides. I mean, we've looked at the effects of individual peptides, and I think the interesting thing is we can find that these peptides are co-localized, and there's a co-localization of peptides which are both inhibitory and stimulatory. So how that release is regulated when you've got peptides in the same nerve cells, presumably released at the same time, and how they're released and differentiate between whether the gut needs stimulating or inhibiting is currently unknown as well.
Okay, thank you very much. And second question, concerning real-world applications. How do you think we should deal with these peptides concerning selectivity of these compounds integrated in some products that we can use as insecticide? How can which are the effects on non-target insect or non-target invertebrates?
Yeah, I mean,
how can you manage this?
I guess that's that's the same with every pesticide application, is how to target the pest, not the non-target species. I mean, peptides are claimed to have some selectivity, but I mean, clearly a lot of these peptides are present in most insect species, and there is a lot of cross-reactivity between the species. We have actually done some work with diuretic hormones and characterized the receptors from Drosophila suzuki and from the honeybee, and we find we can find a differential activity between the peptides on the two different receptors. So we find that the diuretic hormone peptide from Drosophila suzuki doesn't activate the diuretic hormone receptor in the honeybee, but the honeybee diuretic hormone activates the fly receptor. So more work needs to be done looking at the selectivity, but there is an opportunity here that it may be we can find some peptides that are selective for a group of insects and not others. So if we can find something that differentiates between the target species and a non-target species, all the better. But you know, it's going to be very difficult to be that specific. The information that we've generated so far, it looks like there's little cross-reactivity, at least between mammalian peptides and insect receptors. But of course, the reciprocal needs to be done as well. So we need to look at the activity of insect peptides on mammalian receptors.
And this is just a curiosity, but are there differences between heterometabolous insects and heterometabolous insects? Maybe this could be a sort of filter.
Yes, I mean, if you look at some of the in some of the genomes, and I can't remember off the top of my head what the differences are, but there certainly is some differences between insect species where some peptides and receptors seem to be absent in some insects and not others. So again, they could be targeted as in the pest species.
Okay, then I see some questions in the chat. Sherina Amib requested your presentation. She can have the presentation. And then there is Stijan van den Brander that wrote, "Thank you for the interesting and clear talk. As you mentioned that they pose potential targets for insecticidal compounds, I wonder how conserved this system peptide receptor is between insects. If it is conserved, how do you overcome to not target non-harmful beneficial insects?" It's more or less the question that I...
Yes, which basically I guess I've tried to answer. I mean, there may be an opportunity of how you deliver the peptides as well, but you know, there may be some opportunity to be more specific in the delivery, but yeah, at the moment, I mean, that's something that we need to consider.
Okay. So, thank you very much, Neil. And
Yeah, thank you.
I leave the floor to my colleague, session chair, Kristopher Giles. Please.
Thank you. Yes, what an interesting set of talks so far this morning, so diverse. And so our next speaker is Dr. Claudio Lazzari, and he is a professor at the University of Tours, France, and also Buenos Aires, Argentina. And he has spent several decades studying hematophagus organisms, particularly insects, and in particular, vector biology studies, insect physiology studies, neurobiology, and sensory ecology. So a broad range of work. And he's going to talk about today about adaptations, evolutionary adaptations of insects in the ocean and some unique aspects. So, Dr. Lazzari.
Thank you very much. I want to start thanking the organizer of the meeting and the session for giving me the opportunity to share with you some ideas and results concerning a very old question in entomology that still remains unanswered, and is why are insects so rare in the ocean? And as you know, insects are among the most successful organisms in ecological and evolutionary terms. They have colonized almost every habitat in mainlands. So in terms of diversity and biomass, they are extremely abundant. Yet this success in the mainlands contrast with their virtual absence in the oceans, which constitute the most of our biosphere. So this question, there are some species that have succeeded in inhabiting oceans in particular ways. The best known example are ocean escapers of the genus Allobates, the Garidae, who live at the surface of the ocean. So the points here in this card shows the distribution of different species. So they are actually present in the open ocean, but they just move on the surface. So they don't go underwater. So they cannot be considered truly oceanic insect. The reasons for this absence are still a matter of speculation. Several ideas have been posted, and this is a subject in books on insect ecology. And most of these arguments trying to justify this absence are related to the morphology and particular physiology of insects that could render them incapable to survive to the particular conditions of the oceanic realm. Yet there is a small group of insects that challenge these ideas and can help us to understand to what extent insects are limited to occupy this environment. And these are lice. Lice of the order Thyratodon are small insects, wingless. They parasite mammals and birds, on which they feed on the skin derivatives or blood. And what is most important is that they are hemimetabolous insects whose life is related to a permanent and obligated ectoparasitism. So they only can survive on the host. And this is why lice became a classical model in host parasite coevolution, and the phylogenetic trees are mirrors of those of the host. And in this group, there is a family, a small family composed only by genetically species of sucking lice, so those feeding on blood, that have established a very old association with amphibian mammals, pinnipeds and river otters, who spend a part of their life in the water. In some cases, most part of their life in the open ocean. And what happened with these lice when they go to the sea, if according to the present theories, they will not be able to tolerate these conditions. So to try to understand this, we focus on a particular group of Echidopteridae, ectoparasites from sea wolf and sea elephants. Here is Otaria flavescens, and the pinniped living in Patagonia in Argentina, who spends a long time in most part of the year in the water in the sea, and coming ashore in December for the reproductive season. So they start ashore during January and February from the southern hemisphere. There they reproduce, they give birth to pups, and this is the moment when pups can get infected by these lice. After a couple of months, when juveniles remain on land and they molt, they change the skin. This is another moment when lice can be removed from the body, so the lice need to be reinfest them. And then the seals go to the sea, where they can remain for several months far from the shore and performing deep dives for feeding. Until recently, nobody knew what happened with lice, with the ectoparasites during this period. And the predominant idea was that lice should die when the hosts go to the sea, or remain in a kind of latent form, waiting for the return inland of the host. But a couple of years ago, one of the authors of this presentation, Maria Soledad, working in Antarctica, found adult seals infected by adult lice, meaning that the lice were attached to the core, to the body during a long time, even during the excursion on the ocean. So we decided to study the system to see to what extent these lice will be tolerant to the particular condition that they will found in the ocean due to the amphibian way of life of the host. And we start collecting lice in the host. So these lice, you cannot rear them in the lab, you can keep them in the lab for a couple of weeks only. So you need to go to the field and recover them from the belly and flippers of sea lions, trying to not stress a lot the host. So we made some basic experiments in the lab, exposing different stages, eggs, first nymph, and advanced nymph and adults to different conditions of temperature, submersion or exposition to air, and water containing a normal amount of oxygen or hypoxic seawater to test to what extent they could survive. So what we observed that eggs do not tolerate the exposition to low temperatures, similar to those that you can have in the Southern Atlantic, where they live. And also they do not survive underwater. They die immediately. If we look at the nymph and adults, the first things we can observe is that the contact with water triggers akinesis. So they become immediately immobile. They stop moving at all. They keep attached to the substrate if they have, but movements are stopped. And also that when we compare different developmental stages, first instance nymph only survive for a couple of days, whereas advanced nymph adults in our laboratory conditions succeed to survive much longer. So we see that eggs and for instance nymph do not tolerate the conditions that they will confront if the host go to the sea. Yet they are only found in non-swimming and non-molting pups. There is a short period of about a month when the pups stay ashore. They don't go to the water and they still don't molt. And the tolerant stages are only found in adult hosts. This means those that go to the sea for long excursions. Concerning the amount of oxygen in the water, we found a significant difference in survival of lice exposed to normoxic or epoxic water, suggesting that they could have a mechanism for exchanging oxygen with water. Another extremely important factor to which they are exposed during diving excursion of the host is hydrostatic pressure. So the deeper you go, you experience an increase, rapid increase in the hydrostatic pressure that can crush the body of the insect. So to evaluate the tolerance to hydrostatic pressure of these lice, we choose this time lice who are associated to the amphibian mammalian that goes deeper, has records in diving, elephant seals. Elephant seals can dive beyond 2,000 meters. That is an incredible pressure at this depth. Elephant seals show particular adaptations to support this condition, and they can go in the deeps during long time, up to 2 hours. So during this time, the lice that scored them are exposed to exactly the same conditions of hydrostatic pressure and lack of oxygen. This is the species that we studied, Leptothyrus macrorini. This is another species. This is present on this group. You can see here the claws, allowing them to grab to the fur, to the skin of this host that which skin, the fur does not allow the formation of bubbles. There is no air bubbles retained inside. So the lice, the ectoparasite, are directly exposed to the condition of the water. We collect some of them. We brought them to the lab. You can keep them alive for about 2 weeks and perform different experience of tolerance to hydrostatic pressure, simulating the conditions that they could experience when the host dive at different depths, 300 meters, 800 meters, 1,500 or 2,000 meters. And accidentally, we have exposed one of them to a hydrostatic pressure of 450 kilograms per centimeter square, which correspond to 5,500 meters depth, and it survived. As you can see here, adults survived 100%. They were not disturbed for this. Of course, they remain immobile, but they tolerate this pressure. Nymphs were a bit less tolerant, but in any case, the scores were very high. And to confirm these results, we made subsequent exposition. We exposed different groups of adults and nymphs twice to high and low pressure to evaluate for physiological responses, adaptations to tolerate or reactions to the stress produced by the first exposure. And the survival was equally high, and the recovery time, so the time they took to break this akinesis state was very rapid. So in less than 1 minute, in most cases, only in nymph have taken more time to awake, to recover, but in any case, the majority was alive. So meaning that they are able to support this condition. So the next question is how? What are the adaptations allowing the insects this incredible performance? So the first thing we can do is looking at those stages that do not support immersion and those stages that do support. And in an evident difference is the presence of scales, so which are absent in young nymph, but present in advanced nymph. And the presence of scales could be related or could help to the survival underwater. In which way is not clear yet. The presence of these scales and the evidence that stages with scales supported better submersion has been noted several years ago, and two hypotheses have been proposed. For Murray, for instance, scales could provide mechanical protection and protection against desiccation, whereas Hinxton proposed that scales could form a plastron. Plastron is a physical gill that is present in many insects. So you just need microsculpture able to keep a small layer of air, able to exchange gases with the water. We don't know yet if this is the case. We are working on it. And actually, these first results arose a lot of questions, alternative questions deserving to be answered, and on which we are working at this moment. One is there is actually aquatic respiration. We have now tools to evaluate oxygen consumption. We are waiting for the season to test this possibility. Do they have a plastron? This is a question that should be answered mostly performing morphological studies. Is there a metabolic arrestment when they are underwater? This akinesis that is triggered by the contact with water may think that what the insects are doing is reducing the energetic expenditure by reducing the metabolism, and answer that can be obtained also measuring metabolic variables. We don't know what happened with the spiracles. As you know, the tracheal system is filled from air. Air is a compressible medium. So immediately when the insect dives, the internal pressure should make the air inside the tracheal system to go out, but maybe that the spiracles are closed, so they keep this air as a storage. The function of the scales remains still open. It may be that they possess, as many other insects, respiratory pigments, serving to store oxygen associated to particular molecules, and that will be available when the animal is underwater. How they protect for the stress? The stress is not only hydrostatic pressure, there is a thermal stress, low temperature, there is an osmotic stress. Also, there are many factors that could trigger the synthesis of molecular preparatory systems like the heat shock proteins, or the synthesis of piezolytes, which is molecules that protect the integrity of the cellular membrane against hydrostatic pressure, the increase of pressure, and the other. Summarizing, we can say we know for previous work the seal leaves have a terrestrial origin, just as they host. They have associated to pinnipeds when they were still terrestrial animal with the ancestor of pinnipeds. And apparently, all these specialized traits together with host specificity suggests that seal lice co-evolved with their host, so they went together to the marine environment, to the sea. And it is clear that seal lice don't just die or remain ashore, they score the hull, so they actually experience the extreme conditions present that we can find in the deep ocean. So the particular association on the host is obligated association and very old coevolution of the ectoparasite and the host had gave place to adaptations allowing them to support this way of life, the particular way of life of the host. That they only expose a limited time window to reproduce. They can only reproduce when the hosts are ashore and before the pups cool molt. And for some species, it's about 1 month. This is very short. That means that they can have only 1 generation per year. And reproduction should be synchronized with that of the host. But what is most interesting in this context is that we didn't found any particular adaptation or something that make these insects extraordinary or different to other insects, and beyond the extraordinary performance that they show, the endurance to extreme conditions. So in any case, we can explain this extraordinary endurance just with the proper combination of traits that we can find in other insects. So insects are not naturally incapable to survive in the ocean. So we need to look for reasons of the absence in other places, trying to found other reasons making them so rare. So if we come back to the original question, why are no insects in the ocean? So we can say, we can reformulate, it's not no insects, so few insects in the ocean. And the reason could probably be found analyzing the ecological constraints, so the ability to compete with crustacea or to escape from predators, or as had been proposed in their evolutionary story, they could co-evolution with angiosperms, even though there are marine angiosperms, they are pollinated by crustaceans, so they take the place of the insect. And also the aquatic origins of insects that will make for some others challenging to go the way back to return to this. So I want to thank you for your attention, and also the agencies that made possible to this work, NRAE, the Company of Biology, and the Argentinian Agency for Scientific Research, and also to our collaborators that work with us, that made this work possible. And I invite you to see the extended abstract of this work in the site of the conference. Thank you very much.
Thank you, Claudio. There are some questions that we're going to get to in the Q&A, but I do want to ask you a quick question, and it has to do with the idea of a control group, and it's in particular surviving pressure. So are there any studies on some terrestrial lice that indicate that they can survive these pressures that are similar to those that are on elephant seals? So are they pre-adapted? Is the body form pre-adapted in some way?
There are some studies, but with a different focus. There are some work trying to head lice by submerging them underwater. So to see if one can get rid of lice if we go snorkeling or diving. So this is the only kind of work. And the only factor has been evaluated the survival. And of course, they have been done in shallow water. There are no experimental data, to my knowledge, where other species, Vediculus, for instance, Vediculus humanus, or other, had been exposed to high hydrostatic pressure.
And I have two other questions. I'm not sure if you can see those, but I will ask those by Dalton. Are there insects that spend all of their life in the water? And of course, there are those in freshwater, but I think we're thinking marine in this case, right?
In freshwater, there are insects that can develop this plastron very early, so they never go on the surface. These are a couple of species. There are other species that recover air bubbles on the surface and they dive, and others that have extraordinary adaptation to survive in salt lakes or deep lakes, migrating vertically. But in seawater, to my knowledge, there is no example of insects living the whole life under marine water, underwater. Allobates, they do this. They reproduce in debris island on the ocean, and they surf in the surface, but they don't go underwater.
And specific to the actual questions related to the talk, do the lice on elephant seals, do they carry disease?
That is a very good question. Actually, there is an alpha virus that have been isolated from lice in the Macquarie Island, the south of Australia, that could be pathogen, but it's not clear. The presence of a virus doesn't indicate necessarily a pathology, but they host, or they are potentially potential vectors of practical viruses, for instance, and other pathogen. But up to today, I don't know. I don't know diseases but the possibility is open. There are some studies that analyze this possibility.
Well, I know there's several other questions that you might be able to type in the answer afterwards. But thank you, Claudio. It was very, very interesting. Appreciate it.
You're welcome.
Okay. And I don't think Blas has joined us, so I'm going to introduce our next speaker. Anđelko Petrović is our next speaker in this group, and he spent much of his career studying systematics, and in particular, aphid parasitoids, and a broad range of work in IPM of aphids. And so I've seen his publications for many, many years. So it's really exciting to hear him talk. And I know today he's going to talk about, I think, some ongoing issues that all of us have trying to resolve the taxonomic groups of the organisms we work with. It may seem that it would be quite simple with aphid parasitoids, but it's clearly not the case. And so Dr. Petrović is going to talk today about that particular topic, in particular aphidionase. Dr. Petrović.
Thank you very much for the introduction. We heard a lot of interesting talks today, and most of them are state of the art and using cutting edge technologies, but I will try to bring us to the basics. And first I will share my screen. I will talk about taxonomy and systematics, and actually how big is our knowledge gap in taxonomy and systematics. As we all are aware of, biodiversity crisis exists for decades, and just recently it actually started to get public attention that it deserves. Actually, the last year on World Economic Forum, it was emphasized that biodiversity loss is one of the most important threats for the humanity. So there is an urgent need to mitigate this crisis by the ways of exploring, managing, and conserving biodiversity. And the very first step in that mitigating that crisis is actually to overcome the ignorance from policymakers most of the time, and to realize that the most important unfinished job in biology is actually discovering and describing biodiversity in order to protect that. And that brings us to vital role of taxonomy in today's biology, in my opinion. And as you all know, taxonomy is a discipline with a task to describe and name organisms, and actually it represents the bridge between morphology and systematics. And all of those disciplines are actually the basic for all other biological disciplines. So taxonomy is not the highway bridge that we need, because we need a highway bridge to mitigate the biodiversity crisis. But nowadays, unfortunately, taxonomy is more like this bridge on the right. It is an old bridge, decrepit bridge which stands on the good foundation, but with a lot of holes, gaps, and obstacles that need to be filled and fixed. So all these obstacles in taxonomy are actually called taxonomic impediment, and it consists actually from the gaps in taxonomic knowledge, lack of taxonomic infrastructure, and an insufficient number of taxonomists, which are unevenly distributed in the world. And that is actually our reality. So, as we all know, the insects, there is more than 1 million described species of insects, and they represent half of all known organisms on the earth. And it is estimated that the real number of the species is something about 5.5 millions. That means that 80% of the insects still waiting to be discovered. And because of those numbers, actually the insects represents the biggest portion of taxonomic impediment at this moment. In all the insect groups, there are knowledge gaps in taxonomy and systematics, and they just differ in size. And in general, the knowledge gap is negatively correlated with body size of insect. That means that bigger insects we have a lot of knowledge about bigger insects, but we have a very limited knowledge about smaller insects. And also it is positively correlated with numbers of species within groups. Groups with smaller number of species has a small knowledge gap, while groups with high number of species has a very large knowledge gaps in taxonomy. And those are especially this problem is especially big in small Diptera and small Hymenoptera, especially parasitoid Hymenoptera. So one subfamily of insects, subfamily Aphidinae, which belongs to family Braconidae, which are Hymenopteran parasitoids, as an example of how big is our knowledge gap. Subfamily Aphidinae consists of species which are solitary coinobionts, coinobiont endoparasitoids of aphids. That means that a female wasp lays her egg into the live aphid, and aphid continue to grow and feed all the way, all the time. Parasitoid larvae are developing inside the live aphid, and just before pupation, the host aphid dies. And because of this part of their biology, several species of Aphidinae are used as biocontrol agents worldwide to control different species of pest aphids. So they are a well-studied group, and they are considered to be one of the best studied parasitoid group at this moment. And they have a very long tradition in taxonomy. It is the start of Aphidinae taxonomy is actually the start of taxonomy, because the first species was described in 10th edition of Systema Naturae by Linne. And in those 263 years, a lot of notable entomologists actually contributed to Aphidinae taxonomy. For example, Mies von Essenbeck, Halliday, Ashmed, Forster, and many others. But the real pioneers of the Aphidinae taxonomy and systematics are actually our contemporaries. Professor Manfred McCauer from Simon Fraser University in British Columbia, and Dr. Petr Stary from Institute of Entomology in Jelić. Those two researchers started their careers in, I think, late 50s of the previous century, and they are still active. They are retired both, but they are still active, and they publish numerous papers about Aphidinae. And in my personal opinion, Dr. Petr Stary is actually the man who made the difference in Aphidinae taxonomy and systematics, because he published more than 500 papers on all aspects of Aphidinae biology. So they started their work in late 50s and made the good foundations for the rest of us. And you may ask what have, where is the knowledge gap in Aphidine, as Professor Giles already asked. And I will try to explain where is that gap, just analyzing the taxonomic studies on Aphidine, which are conducted in the last decade, from 2010 until 2021. So the first problem which someone may encounter when start reading about the Aphidine is actually the number of described species and genera. When you start to find the right number of described species, you will find the different values. For example, in a publication which was published in 2012 by Boivin and Associate, there is a 400 Aphidine species classified in 50 genera. In the later study published by Jelić and Associates, there was a 505 species classified in 38 genera, and in the same year, Tyne and Associates says that there is more than 600 species classified in 65 genera. Although I don't want to make the conclusions, but according to my estimate and according to available data that I have, there is prior to 2010, there were somewhere around 500 species which were classified in 55 genera. And now we will look what happens after 2010. After 2010, in these dozen years, in my search, I found that somewhere about 1,800 papers were published which are dealing with Aphidine in some way of Aphidine. And among these papers, to my best knowledge, I found less than 60 papers focusing on Aphidine taxonomy and systematics, and only 37 papers in which new Aphidine taxa are described. That means that only 3% of all published papers in the last decade are actually considering taxonomy and systematics of Aphidine, which represents a very huge taxonomic impediment, because on every taxonomic paper about Aphidine, there is 30 papers about applied biology or applied aspects of Aphidine biology in order to use them as biocontrol agents. And thus there is a big discrepancy about all these things. In this period, 3 new genera were described. Those genera are Choreopran, described by Manfred McCauer in 2012 from New Zealand, genera, and also genera Sergeioxys and Astigmapraon, which were described from Palearctic region. And in addition, 55 new species were described. And that was interesting even for me, because never before I put that all in the one place. And actually I realized that 10% of all known species of Aphidine are actually described in last 10 years, actually in last 12 years, which is actually very big proportion of the known species. And also those 3 genera are something about 6% of all known genera. So those numbers alone are speaking for themselves and say that it is very big taxonomic impact. It is very big knowledge gap about Aphidine, although there is a huge history of research and a huge number of papers which are published every year. We still don't know a lot about these parasitoids. When we look actually the distribution of those newly described species in the last 10 years, we will see that 67% of described species, which is 37 species, are described from Palearctic region, 9 species are described from Nearctic region, 8 species were described for Oriental region, and only 1 species were described from New Zealand, while from Afrotropical and the Neotropical region, we didn't found any new species in last decade. And this is not actually the real thing that exists in nature. Actually, this is the other aspect of taxonomic impediment. And actually, this is the results of uneven distribution of taxonomic experts, because most of the taxonomic experts for this group are located in Europe and in Palearctic, but most of them are Europeans. And those 55 species were described by 19 different people, 19 different researchers. Among those 19 people, 11 is from Europe, and most of them are located actually in only 4 countries. It is Russia, Czech Republic, Serbia, and Greece. And that is actually the very, very big problem for Aphidine taxonomy and systematics in general, and for Aphidine biodiversity, because all species, almost all species, were described by authors from Europe. Just 7 species are, there is no authors from Europe. And that is actually maybe even bigger problem than the knowledge gap that exists. But taxonomy goes beyond naming the species. It is not just naming the species, and taxonomists have a lot of different roles in our society. So the crucial role of taxonomists in identification of alien species is generally widely underestimated. And that is, in my opinion, very important role and very important part of the work that taxonomists are performing. And on our example, on Aphidine, we can see that prior to year 2000, there were only 5 alien Aphidine species recorded in Europe. And all of these species were intentionally introduced as biocontrol agents. From 2010 till today, 3 new alien species were detected in Europe, and all 3 detected species are actually accidentally introduced, which is very interesting because prior to this, we never had accidental introduction of those alien species. As I said, there is 3 species, and 2 of them are recorded in Europe soon after their official description. For example, Lysiphlebus orientalis, a species which is described from China, when there was a project about biological control of Apis glycine, which make the problems in the States. And they found the Lysiphlebus orientalis as a new species and potential biocontrol agent, and it was described in 2010. And later on, we detected the same species in the samples collected from Serbia, which were collected in 2010 and 2011. So it was in Serbia prior to their official description. The same situation actually is with Aphidius ericaphidis, which were described in 2011 from the USA and detected in samples from Serbia and Scotland collected in 2014 and 2015. When we look through our collections, later on we look through our collection and determine that those 2 species were actually present in the Europe before their official description. For example, specimens of Lysiphlebus orientalis were collected back in 1995, while in Aphidius ericaphidis it is even further in the past, because Peter Starry collected some specimens back in 1965 in French Alps. So the both species were present decades before their official description, but actually we didn't have the right tools to detect them. And all 3 those invasive species were actually detected using integrative taxonomy. In these particular cases, we use the data from the DNA barcodes, from morphology, and from ecology. And actually that became standard in the last decade in taxonomy of Aphidae, in studies of taxonomy of Aphidae. So only when we were able to make this integrative approach, we actually determined the presence of these species. And in my opinion, that is the only right way to do taxonomy studies. Actually, we should use as much data as we can, especially because of using one kind of data can actually make the biggest taxonomic impediment than we already have. Also, we are facing some other problems in taxonomy, and those problem is when alpha and beta taxonomy are in discrepancy. What does that mean? That means that when naming the species and classifying the species actually is very problematic. When you find a new species and try to put them in the some classification scheme, then you came in the problem which is caused actually by superficial description taxonomic impediment, which means that a lot of species description, especially species description from the first 150 years of taxonomy, are actually too superficial for today's standards. And then the only way how we can reduce the knowledge gap and how we can make congruence between alpha and beta taxonomy is actually to perform the revision of whole group of organisms. And in last decade, a lot of efforts we put a lot of effort to make improvements in Aphidae taxonomy and systematics, because the majority of the tribes, sub-tribes in general from this subfamily were revised 30 to 50 years ago, and majority of those revisions are outdated. So I will mention just a few examples. One example is the revision of the world of the subtribe Monoptonina, and that is the only revision of the subtribe level in the last decade, actually in the last several decades. And those revisions actually include all available species and resulted in discovery of 5 new species belonging to genus Monoptonus, and all 5 species are from Nearctic and they are from Canada. Also, there were a lot of other studies, but not on the sub-tribe level, but on general level, and a lot of generally smaller number of species were also revised. And most of these revisions resulted in higher number of species within the genus, but in some cases, some genera are actually synonymized under the other genus. For example, Eoaphidius and Remundiera were synonymized under the genus Aphidius. Also, there were several revisions of the European species of some genera, and here I will talk just about one revision. That is actually a revision of European Lysiflagus species. And this revision is a classical example of importance of revisions in taxonomy and later on in biodiversity research. Prior to this revision in Europe, there were 15 Lysiflagus species, and during this revision, actual number of species were reduced to 13. And by the way, that 4 species were synonymized and 2 new species were described. And that is actually very important by itself, but sometimes quality of those taxonomic information is even more important than quantity. Working on this revision, authors actually realized that only 2 Lysiflagus species actually have descriptions that match today's standards in Aphidae taxonomy. So all other European Lysiflagus species were redescribed together with remarks about their biology and known distribution, and also the identification key for all European species were provided. which is actually, I think, classical example how this approach can allow us and make allow us to live a little bit easier and to make easier identification of those species. There is also several revision of species groups and complexes, and here I will mention only actually only two revisions. One is, and both of the revisions are actually revisions of species which are commercially used biocontrol agents. First species group is Aphidus colemani group, which is probably the most widely used species in biological control, and it is commercially produced as a common biocontrol agent against a huge number of pest aphids, mainly in greenhouses, but in later years, this species is used also in open fields to control different pest aphids. By this study, authors actually realized that this species group is consistent of three known species. All three species were previously unknown. Those are Aphidus colemani, Aphidus transcaspicus, and the surprise was actually Aphidus platensis, which were a species which was almost forgotten. And at the time of the study, actually the most interesting result and most important results for non-taxonomists is actually that at the time of this study, when this study was conducted, parasitoids within those globally commercially distributed materials actually were a mixture of all three species. And I'm sure that situation is very similar at this moment. So probably if you're buying some product that is written Aphidus colemani, you will buy all of these three species. And the second example is actually the Aphidus Ed species group, which is also a biocontrol agent, and it is used worldwide against pea aphid. Pea aphid is known pest of several leguminosa species, and it is very common to use Aphidine parasitoids in control of this aphid pest. In 2019, we published a study which actually showed that this group is also consistent of three different species, namely Aphidus smithi, Aphidus sedi, and Aphidus banksii. And the most important actually result was that species Aphidus banksii, which were described as Aphidus stadi in 1990, if I'm not wrong, it was described from Israel and Turkey, and it was actually part of the biocontrol program of Acetosiphon pisum in North America, and it was introduced in North America under the name of Aphidus smithi, and later on they actually realized that it is not Aphidus smithi, that is a different species which actually lived in Israel and Turkey. Our analysis identified that this species is widely distributed with range that cover most of Western Palearctic. Those blue dots actually are actually samples that we analyzed of the Aphidus banksii, and its range is from the United Kingdom all the way to Israel. So if such huge knowledge gaps in taxonomy of economically important biocontrol agents exist, they raise numerous questions about the taxonomic status of many other Aphidine species, especially those ones which parasitize aphids or non-agricultural pest plants, which are all usually not the subject of many studies. So there is a lot of things that should be done in the future. And for the end, I would like to go back to this bridge from the beginning of the story. This taxonomy bridge looks a bit different after decades of research. Now, some holes are filled with the knowledge, some obstacles are removed. So we can see further, and what we can see actually new holes, new obstacles that need to be fixed. And even with the tremendous work that has been done so far, the knowledge gap in Aphidinae taxonomy is still very big, and all aspects of taxonomic impediment are more than obvious, especially one with uneven distribution of taxonomic experts and small number of taxonomic experts. And I would like to conclude with that there is no meaningful research in biology without reliable taxonomy. And we see in previous talks, especially when we're talking about mosquitoes and malaria, they actually try to work some state-of-the-art molecular taxonomy on Anopheles mosquitoes, which was very interesting. And also the Fabrizio was talking about some parasitic wasps, and two of those wasps are actually Aphidinae. They are Aphidius cervi and Aphidius and Lysiphlebus fabarum, and both of those species which have sequenced the genome are actually taxonomically problematic species, which are not, we are not clear yet is it one or more species in that group. So as I said, there is no meaningful research in biology without reliable taxonomy. And in addition to that, Swiss Reinsurance Institute last year actually published the one research in which they stated that 55% of global GDP is moderately or highly dependent on biodiversity and ecosystem service, and that at least half the countries of the world lost at least 1/3 of their ecosystem service because of the loss of biodiversity. Even if we know all that, and even if we know all that for decades, importance of taxonomy and systematics in this world, in modern world, is far from being fully acknowledged, and we are still lacking large research grants for taxonomy. And in developing and undeveloped country, we are lacking even the small grants for taxonomy. And until institutions, governments, and the world realize the importance of this kind of research, we need to be clever and utilize the data from all available resources that we already have, such as museum collections, molecular data, ecological data, and combine all of them to perform actually our research. The one who would like to take this interesting walk over the bridge called taxonomy must be aware of some things, and those things that if we use one type of information, we can make the situation even worse. Actually, we can make the knowledge gaps even bigger. So in my opinion, and in particular case for Aphidinae, the taxonomy must be integrative, which will use all available data, of course, using the barcodes, which are very useful, but their barcodes alone are actually not enough because of a lot of problematic things with this. I think similar situation is most other taxa, and if this situation is with probably the best studied parasitoid group, and then you can imagine how bad are actually the situation from less studied groups. And it seems appropriate here to finish this talk with a citation of the song by Jonathan Colton, which I adapted a little bit for the taxonomy. So we have experiments to run, there is research to be done on the species who are still alive. Thank you for your attention.
Thank you, Angelko. Can we have any questions from the audience?
There is one on the Q&A box.
Are you able to see that question?
Yeah, I can see. I would like to ask about your estimate of species and general numbers. Do you count only recent taxons or fossil too? I was talking about just the living species. No, I didn't count the fossil species. With the fossil species, there is probably now somewhere about 600 species, and as I see, this is Marina. So she actually described several fossil species in the last year, I think. So, but focus of this talk was actually on the living species, not about fossil species.
I do have a quick question for you. Your epilogue is, of course, unnerving for us who have studied the ecology of parasitoids and made bold conclusions. I mean, I guess in some ways, the discovery of species complexes is concerning. Maybe that could only be resolved with voucher specimens and looking back and backwards. I mean, that's part of what we're going to have to do. But with the number of studies, was it 95% on the biology and ecology and 5 to 10 on taxonomy? Well, we have some unresolved biological conclusions, maybe.
Yeah, we have. And I think the also very big problem is that a lot of type specimens are missing for a lot of groups. So that is also the big problem.
Yeah.
And especially the species which were described in the 19th century, Holiday, Knees, and other great entomologists described a very few sentences about every species. So that is very, very problematic if we don't have the type material.
There's another question there.
Okay. Do we have any project that investigates Aphidina species in the world where other countries can collaborate? Actually, there is no specific project, but we can collaborate on any part of the world. Actually, we are working on all fauna of Aphidina. So recently we published a paper about Aphidina of Kyrgyzstan from the Linz Museum collection. So if anybody has any idea for collaboration, you can write me on my email. Do not hesitate, please. And the second one is, to what extent may evolution and divergence be impediments to taxonomy if they can? Oh, that is very, very hard one. Yeah, they can be impediments to taxonomy, but I think that we are facing taxonomy impediment, which is actually impediment for ecology and biodiversity research, which is much bigger problem than those divergence in taxonomy itself. I think that we need to accelerate the taxonomical studies in order to provide relevant data to ecologists and all other actually biologists. And there is another one. What is your idea how we taxonomies can survive in the age of biodiversity or just expire? I'm not so pessimistic. I think we can survive because there are some things that start to change. For example, German barcode of life project, GVOL 3, I think. They actually make the new working places for new taxonomists, especially for Diptera and Hymenoptera, which have the biggest proportion of dark taxa. So I hope that developed countries will realize, all developed countries will realize the importance, and that other will follow. Until then, we can just use actually the projects that are not clearly taxonomic, but more applied, and then work out taxonomy in between.
I want to, there's several other questions. Maybe you could answer those electronically. I know we are a little over, so Nick or
okay,
I don't know how much further we should go at this point. Oh, Cedric, yes.
Yeah, just go ahead.
Okay, great.
Okay. The next question is, in developing country taxonomies are rare. Anything you can do to discover species, especially in those countries in the tropic. I'm not sure that I understand the question, but we can always cooperate between each other. That is the only way that we can actually make the progress. Cooperation is the only way. Yeah, I know that taxonomists are rare, and even in in developing countries, and they are actually rare in developed countries. So there are just a few institutions in every country which have a lot of taxonomists. And the last question is that barcodes are not enough for taxonomy. It needs integrative taxonomy now. Yes, actually, that is my opinion based on my group and probably your group. I know that because I know the group. So in my group, that is not enough. Maybe there are some groups which can be used, but that is not a situation. Okay, thank you very much. Can I abuse a little more time as a session chair also? I would just like to thank you to Journal Insects and to organizing people, and to Cedric for this session, and also to Nickolas, who make all these things possible together with Barbara. Thank you very much.
Thank you, Angelico. So I guess not just a few more words from my side. I would like to thank the conference chair, the session chairs, and the speakers for their time and interesting contributions to this first live session of IECE 2021. Also, a thank you to our participants. I hope you found this webinar interesting. As a reminder, the recording of the webinar will be made available as soon as possible on the conference website. So thanks again to all of you. I wish everyone a nice day, evening, wherever you are in the world. Have a good weekend and take care. Bye.